April 12, 1937, a day that began like any other in the bustling streets of New York City, yet would end with more than 100 innocent lives extinguished by a single lethal cure. In a cramped laboratory on the Lower East Side, a chemist mixed powdered sulfanilamide with diethylene glycol, an industrial antifreeze, and called the resulting concoction Elixir Sulfanilamide. It was marketed as a miracle cure for everything from rheumatism to the common cold, and within weeks it was being sold over the counter in pharmacies across the nation. The price was low, the promise high, and the danger unthinkable. To understand how such a tragedy could unfold, we must travel back to the early 1930s, a time when antibiotics were still a distant dream and physicians relied heavily on patent medicines that were barely regulated. Sulfanilamide itself was a breakthrough. Discovered in 1908, it was the first synthetic antibacterial agent, and by the mid-1930s, it had saved countless lives from deadly infections such as streptococcal pneumonia. The drug was water-soluble, but it did not dissolve well in plain water, a problem that drug manufacturers were eager to solve. Enter Dr. Francis M.C.L. McIntosh, a young chemist working for a small pharmaceutical firm known as the Miller Laboratories. Faced with a growing demand for an oral form of sulfanilamide, McIntosh turned to diethylene glycol, a sweet-tasting, clear liquid already used in industrial cooling systems and as a humectant in cosmetics. The solvent was cheap, readily available, and crucially, it masked the bitter taste of the drug. In a single batch, McIntosh mixed 5,000 milligrams of sulfanilamide with 250 milliliters of diethylene glycol, bottled the solution, labeled it Elixir Sulfanilamide, and sent it out to distributors. The marketing campaign was aggressive. Newspaper ads promised swift relief from fever, cough, sore throat, and skin eruptions, while doctors' brochures extolled the elixir's miraculous healing power. Because the Food and Drug Act of 1906 required only that labeling be truthful, and because there were no mandatory safety tests for new drug formulations, the product slipped onto shelves without any clinical trials or toxicity studies. Within a week of its release, families began reporting strange symptoms: sudden vomiting, severe abdominal pain, and a rapid decline into coma. In New York City alone, hospitals recorded a surge of patients, young children, pregnant women, and the elderly, all presenting with the same baffling constellation of signs. By the end of May, the death toll had climbed to 107, and the nation was gripped by horror. The first autopsies were performed at Bellevue Hospital, where pathologist Dr. Edward J. H. Hill led a team that quickly recognized the pattern. Gross examination revealed swollen, hemorrhagic kidneys and a frothy, edematous liver. Microscopic analysis showed tubular necrosis and widespread cellular death in the renal cortex, a classic picture of acute renal failure. Blood chemistry, still a fledgling science, indicated a precipitous rise in serum urea and creatinine, confirming that the kidneys had been catastrophically damaged. The culprit, however, remained hidden. It took a chance observation by a pharmacist in Manhattan, who noted that the elixir's sweet taste was unusual for a sulfanilamide preparation. He sent a sample to the U.S. Public Health Service, where chemist Dr. George B. L. Stokes identified diethylene glycol as the solvent. Stokes, aware of the compound's toxicity, recalled that exposure to even modest amounts could cause severe renal failure, central nervous system depression, and death. The chemical's LD50 in rats was a mere 2.5 milliliters per kilogram, a dose far lower than that ingested by the patients. Armed with this evidence, the New York County District Attorney's Office filed criminal charges against Miller Laboratories and its owner, Mr. William C. Miller. The trial, held in the summer of 1937, became a national spectacle. Prosecutors presented the grim autopsy photographs, testimony from grieving mothers, and expert witnesses who explained the pharmacology of diethylene glycol. Defense attorneys argued that the solvent was a common industrial ingredient and that there had been no intention to harm. They claimed ignorance, insisting that the chemist had followed the standard practices of the day. The jury, after 2 weeks of testimony, returned a guilty verdict on charges of criminal negligence and fraud. Miller Laboratories was ordered to cease production, its assets were seized, and Mr. Miller was fined $5,000, a sum that, while substantial at the time, paled in comparison to the loss of life. The court also ordered the company to publish a retraction in every newspaper that had run an advertisement for the elixir, a rare judicial remedy meant to inform the public and prevent further tragedy. The fallout rippled far beyond the courtroom. Public outrage forced Congress to confront an obvious flaw in the nation's drug regulatory framework. There was no requirement for manufacturers to prove a product's safety before it reached consumers. In response, President Franklin D. Roosevelt signed the Federal Food, Drug, and Cosmetic Act into law on June 25, 1938. The new legislation mandated pre-market safety testing, required accurate labeling of all ingredients, and gave the Food and Drug Administration the authority to enforce compliance. It also established a system of mandatory reporting of adverse drug reactions, a direct legacy of the elixir sulfanilamide catastrophe. In the years that followed, the medical community revisited the autopsy findings, using them as a cautionary tale in pathology courses. The incident sparked a wave of research into nephrotoxins, leading to a deeper understanding of how certain chemicals disrupt renal tubular function. Subscribe to the Autopsy Files for more stories like this.